Alright guys, welcome back. So one last video for this unit, and for this video we're going to talk about our humoral immunity. So that's the last one we need to talk about to end this unit. Now when we're talking about humoral immunity, we're not talking about our cell-mediated anymore. So remember, cell-mediated was when we were talking about dealing with cells that were infected with a pathogen or an antigen. In this case, we're going to be dealing with not cells, but pathogens and antigens that are present in our bloodstream. Okay, so in humoral immunity, one of the major things we will be dealing with are proteins called antibodies, and we'll talk about those in a couple of seconds. For us now, humoral immunity, if you're immune to something or to a particular pathogen, it's because you have those protective antibodies already in your immune system. So let's get right into it right now. So here is a look at our antibodies, and if you see antibodies in a textbook, you'll always see them, or pictures, anything like that, you'll always see them drawn in a Y fashion. Now, that's because most of them have this Y shape. Oftentimes, you'll see them abbreviated as IgG, which is short for immunoglobulin, which is the type of protein that it is. So an antibody, immunoglobulin, means the same thing, okay? The antibodies are produced by the B lymphocytes. So we talked about T lymphocytes before. Now we're going to be talking about B lymphocytes. And antibodies are very specific, and they bind only to one antigen. Okay, so we'll have antibodies for any pathogen we've ever come into contact with, and it'll be specific for that particular type of pathogen. It won't be for anything else. So you don't have antibodies that deal with 3 or 4 different infections. You have an antibody that deals with one pathogen at a time. So why are they always drawn like Ys? Because they have, like any protein, they have a shape. Antibodies are made up of different protein chains. We get 2 heavy chains. And in the picture here, this is going to be the purple ones here and here. So those are our heavy chains. And then we also have these 2 yellow ones, which we call the light chains. There are disulfide bonds that connect all of these things together. So where you see these bridges here, those are going to be your disulfide bonds. Now, right at the tips of the top of the Y, you're going to have 2 regions that are going to have 2 similar names. So these areas are going to be the variable region, and then right at the very tips up here, you're going to have the antigen binding site. The reason why these are variable is because the shape of this is going to be different for each antigen, and then the different shape is going to allow them to bind to all of these different antigen. So if we look down here at this picture, you can see the antigen binding site for this antibody has got that V-shaped, which means it's not going to fit on this square-shaped part of this antigen. So the stem and the constant region, so the constant regions are these areas right here. Those are going to stay the same for each antibody class, and there's several classes of antibodies. We're not going to get into it in this course. So they would stay the same, and then the variable regions would change so that they could bind to each different antigen. All right, so where do these antibodies come from? How do we produce them? Well, they come from our B cells. Now, our B cells will have antibodies on the surface of their cells. So if we look at our cell down here, let me just turn on my pen. We'll see antibodies on the surface of that B cell. So the B cell will have an antibody, and the antigen will attach itself to the antibody, and then that antigen can be brought inside of the B cell. Once it gets inside the B cell, it's processed, and it turns itself as a signal on the outside of the B cell. This B cell will travel around until it comes into contact with a T helper cell. So our T helper cell will find this B cell, it'll recognize the antigen being displayed. It'll release a bunch of these cytokines that we talked about in the last unit, which are chemical messengers. And those cytokines will get that B cell to turn into a different cell called a plasma cell. And what a plasma cell is, it's basically a factory for producing antibodies. So you can see here, this plasma cell starts to spit out all kinds of these antibodies, and those antibodies are going to be specific for that antigen that we picked up right at the beginning. And that's generally the really short way about how our antibodies are produced using B cells and helper T cells, plasma cells, and then finally producing antibodies. Alright, so once these antibodies are produced by the plasma cells, then they're free to go through our blood plasma. And they're going to travel all around our body, and they're going to look for pathogens or specific antigens that they recognize. So that can be a bacteria, it can be a virus, it can be a fungi, it can be anything like that. And their objective is to find a pathogen. In this case, in the example right here, is going to be a virus. And then here's our antibodies, and they'll attach themselves to that pathogen. Now, in this case here, you can see there's so many antibodies around that virus is not going to be able to even touch that host cell, so it won't cause infection. So the function of the antigens is to, the antibodies is to prevent infection, to remove, disable, or destroy an antigen or pathogen, and help to enhance phagocytosis. So that's the general functions of these antibodies. Now, antibodies have a number of ways of being effective, and these are 3 examples here. Number 1, we talk about neutralization. And neutralization is like the example we just talked about in the previous slide, is when an antibody recognizes an antigen, and we collect so many of the antibodies around there that we basically make that virus incapable of coming in contact with a cell. So if the virus can't actually touch the cell, it can't find the cell receptor, it can't get inside, it can't be effective. So neutralization is just basically surrounding the virus until it becomes inactive. The second one here we've got is opsonization. Opsonization down here is basically an antibody. They come along, they label the antigen with special antibodies called opsonins, and then that makes the antigen much more attractive to phagocytic cells. So opsonization is a way of enhancing phagocytosis. It makes it easier for phagocytotic cells to find antigens and to get rid of them that way. And then finally, antibodies are able to activate the complement system that we talked about when we talked about our innate immune system, the non-specific immune responses. So that group of 20 large proteins that help with inflammation, phagocytosis, lysis, they also do some opsonization of their own. That's another way that antibodies are effective against a pathogen. So neutralization, opsonization, and activation of the complements. Okay, just like our cytotoxic T cells that needed to be activated, our B cells need to be, or B lymphocytes, they need to be activated as well. And one of the ways that they're activated is by either direct recognition of the antigen, like we showed in the example before that, or they can be activated and turned on by T helper cell too. So this shows the importance of the T helper cell because it can help connect the cell-mediated response with the humoral response. So the T helper cell is a very important cell in that respect. Now once those B cells get activated, they can undergo cloning again, and those clones can then go on to produce plasma cells, which are the cells that will produce our antibodies. This cell here is kind of talking about clonal selection and talking about how both sides of that specific immune system works. So once cells become activated, they produce what we call effector cells. Now effector cells are going to have an immediate effect. They're going to be short-lived, and they're only going to happen, or they're going to be around while we're sick. After we're sick, those cells tend to die off. So when we're talking about effector cells, we're talking about active helper T cells, active cytotoxic T cells, and those plasma cells that are producing antibodies. Now, one of the major aspects about our immune system that makes it so effective is that we have memory. So one of the things that these cells can also differentiate into are these long-lived memory cells. And we'll have T cells that we already talked about, memory T cells, but we also have memory B cells. Now, this is where our memory comes into, the memory part of our immune system becomes very, very important. Our primary response is what happens when we first get exposed to the antigen. And if we look at this graph, you can see antibody concentration is on the y-axis, and then we get time on the x. Now, keep in mind that these are two different graphs, so if we put a line in between here. Our primary response, you can see you're exposed, the antibody concentration goes up, it peaks, around right here, and then you get better, and then the antibody concentration goes down. That's our typical response. Now, you can get quite sick in this response, depending on how effective that virus is, or how effective that pathogen is at causing illness, how virulent that pathogen is. Now, the second time you find that pathogen, the second time you encounter that antigen, what's going to happen is, since we have our memory cells, we're going to skip a whole bunch of steps in our immune response, and we're going to go right into releasing antibodies. So you can see in a very short period of time, we have a massive amount of antibodies in our system, and these antibodies will quickly get rid of the infection, the pathogen, and you probably won't actually even feel sick because the response is that quick. And that's because we have these memory T cells and memory B cells. They live a lot longer. And they have a much bigger response when they're activated, and we move in from an initial or a primary response to a secondary response to the same antigen. Okay, this figure right here is quite important for you guys. You should be able to put labels on this if you've got a good understanding of what we talked about in this unit. So a good test question would be for me to give you this, and you should be able to label this and then understand what's going on through each step. So on this side of the figure, we've got our humoral immune system, our immune responses. So this is going to have to do with production of antibodies, right? Over here is our cell-mediated immune response. That's going to deal with cells that are infected. So on this side, we'll be dealing with the B cells. And then, of course, for the cell-mediated over here, we'll be dealing with T. So I want to leave that for you guys to look through. Use this as your kind of worksheet as you're going through the unit to try and understand how all of these systems, how all of these different parts of our immune system come together to do the same thing, which is to prevent infection and to help keep you healthy. The following slide is also a pretty good summary of the humoral and the cell-mediated immune response. So on this column here, you'll have the humoral, and over here, these two columns here will be cell-mediated. And it talks about, what each one is, the name, the functions, and so on and so forth. So make sure you know this, and then if you keep going through the rest of that set of notes, you'll notice that the answers are here to help you along if you're having any trouble filling it out. But I would really suggest making sure, make sure you're quite familiar with how all of this stuff fits together. Thanks a lot.